Alright, so we're going to pick up right from where we left off, Chapter 5. This is the second half of Chapter 5, still talking about some of the weirdo things that we see that are genetically based but don't really follow Mendel's basic rules. More extensions and modifications of Mendel's basic principles. Pages 115 to 125 in your textbook if you're following along at home. And it's also important to note that this begins the material for unit exam number 2. So today we're going to talk about first sex-influenced and sex-limited characteristics. This is different and distinct from sex-linked traits, which we talked about before the exam. We'll discuss cytoplasmic inheritance and how that is gender-based and parent-specific. You'll see what I mean in just a second. Some ideas like maternal effect and genomic imprinting, and we'll end the lecture talking about some of the roles that the environment plays on influencing the phenotypes that we see. So some phenotypes are not completely genetically based. The environment can play a role as well. So we talked about things like codominance and epistasis in previous lectures, and that was weird in the sense that it made Mendel's laws appear to be wrong. It appeared to affect the Mendelian predictions. Now, once we dug down deep and looked at the mechanisms by which these things were working, we could see where Mendel's laws held true. But we had to understand what the DNA and what the genes were doing in order to see that, to see how Mendel's laws were not being broken. So that was weird, but not so weird. The stuff we're going to talk about in this lecture is really weird. As we, and by we I mean the field, scientists, people, even students, as we understand more about genetics, we begin to understand just how little we understand. And that's very powerful to me. I think it's important. I think that's what education brings you, what education gives you. When you're truly educated and you begin to know a lot, it gives you the wisdom and the humbleness to know that there's so much more you still don't understand. Mendel's rules, basic transmission genetics, it's awesome. These principles are relatively simple, they make sense, they give testable predictions that you can demonstrate are correct, they explain and account for the vast majority of the genetic observations we see. But there is more out there that is genetically based but genuinely does not fit Mendel's rules. Even when you dig down deep, you see that Mendel's rules are not holding true. And as rare as they are, these exceptions to Mendel's rules are extremely interesting. Sometimes we know the reason behind the things that break Mendel's rules, but sometimes we don't. And so many of the things we talk about in this lecture, very much at the edge of the genetic envelope, truly observations that we can measure, but we don't understand. So, if some of you are thinking about graduate school and genetics seems to be working for you, there are PhD projects in this lecture. There are things in this lecture that science needs to know and doesn't know yet. So some interesting stuff going on here. We'll talk first about sex-influenced and sex-limited characteristics. We talked about sex-linked traits. The example we used most was color blindness in human beings. And these were traits that showed us distinct gender pattern, and we knew we can measure and track what makes the gender pattern in these sex-linked traits. What makes a sex-linked trait is that it's usually passed from mother to son, and the reason why it's passed from mother to son is because sex-linked traits are X-linked traits. These are traits encoded by genes that can be found on the X chromosome. And since dad gives his son the Y chromosome, that's the gender chromosome, and mom gives the son his only X, X-linked traits become sex-linked traits passed from mother to son. We talked about all that. What we're going to discuss today are traits that are genetically inherited, and they're influenced by gender. Oftentimes they're passed from one parent only directly to the offspring, but they are not traits encoded by genes on the X or Y chromosome. That means these traits are encoded on the autosomal chromosomes, chromosome pair 1 through 22. So the question becomes, how is a trait that is not on the sex chromosomes, but on autosomal chromosomes instead, how is that trait sex-influenced? And the answer is, again, oftentimes we don't know. This is at the edge of our understanding. Sex-influenced traits are defined as traits that are determined by autosomal genes, so they are not on the sex chromosomes, inherited following Mendel's principles, independent assortment, segregation, and all that, but for whatever reason expressed differently in males versus females of the species. Sex-influenced traits can be expressed by both genders. So you can see the traits expressed by both genders, but for one reason or another, the penetrance of the trait is different. In other words, the trait is more likely to be expressed in one gender over the other. We talked about penetrance in a previous lecture. Penetrance is how likely it is that a trait will be expressed. A penetrance of 0 means the trait will never be expressed regardless of the genotype. A penetrance of 100% means the genotype and phenotype are 100% correlated. But penetrance outside of those ranges, between 0 and 100, means that you can have the genotype, but not always have the phenotype. We talked about polydactylism in a previous lecture to drive this home. Here we're talking about gender-specific penetrance, where your gender dictates how likely it is that a trait will be expressed. So in other words, one sex, one gender, expresses the trait more easily than the other. Goats have beards. It's kind of freaky, kind of cool. I'm sure many of you knew that. There's many examples of goats having beards. Oh, I'm sorry about that, actually. I don't know how that picture got there. Typically, a billy goat, that's a male goat, has the beard. So usually when you're looking at a bearded goat, you're looking at a male goat. But female goats can have beards too. It's just that beards are much less likely in female goats than in male goats. So this is a sex-influenced trait, right? Given the definition I just read to you, this trait is sex-influenced. It's genetically based, it's due to inherited genes, it shows a gender difference, and the penetrance of the trait is different in different genders. In this case, the penetrance is higher in males and lower in females. We might not fully understand the mechanism for this, but we do understand the genetics when it comes to bearded goats. If we say or call the allele for having beards BB, and we call the allele B+, that allele which encodes no beard, we see, and here's the sex-influenced part, that the allele BB is dominant in males, but that very same allele behaves recessively in females. So think about that for a second. That's all I should have to tell you for you to understand why this trait is sex-influenced and why it has a higher penetrance in males versus females. We'll go through it, though. Here are 3 different genotypes, the 3 only possible genotypes given these alleles, B+, 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 BB, or BB. Those are the 2 homozygous and the 1 heterozygote. It's hidden behind the little integrity thing there, toolbar there. But in males, the only way you're beardless is if you're B+, B+. That's homozygous recessive for males. For males, the BB allele is dominant, so the heterozygote shows the dominant trait, bearded. And of course, the homozygous dominant shows the dominant trait, bearded. And that's what it says under this little icon here. So it's beardless, bearded, bearded. But in females, you have the opposite dominance pattern. So a B+, B+ female is bearded. Beardless, of course, because that's the beardless allele. But for females, beardless is dominant. So for the heterozygote in females, B+, BB, that female is still beardless because the B+ allele for her is dominant. But if the female is BB, BB, then she only has the information for having a beard, albeit recessive in her genome, but she shows the recessive trait and she has a beard. So if 2 heterozygote goats mate, the male, of course, would have the beard and the female would not in that case because of what we just said. 2 heterozygote goats mate. We're talking about these 2 in the middle. 75% of their male offspring would have beards, while 25% of their female offspring would have beards. Interesting. Interesting. Same genotypes in the offspring, same genotypes in the parents, but sex influence. So let's take that a little further. Let's look at that. So we have the P generation, right? We take our pure breeding B+, B+ male and mate it with a bearded female. Fine, no big deal. We have the heterozygotes here, and this is the mating we're talking about. A heterozygote bearded male mates with a heterozygote not bearded female. Let's do this as a Punnett square because we can. This is a heterozygote parent crossed with another heterozygote parent. We fill in the Punnett square as we've always done, and this is what we get. But when we look at this, we realize that if we're talking about males, for males, the beard is dominant. So for the males, this 75% of the offspring will have beards. This is bearded. Excuse me one second. No, got that wrong. Let me back up for a second. So I want to make sure that this is right. So BB is the beard in males. It's dominant. So The numbers are correct, I just had the genomes wrong. So this 75% of the male offspring will have beards. The homozygote BBBB will have beards, and the two heterozygotes will have beards because the bearded allele is dominant in males. So for males, 75% of them will have the bearded trait. Only this 25% of males will have no beard. For females, the cross is exactly the same. We don't need to change the Punnett square. This is the cross. But for the females, the bearded allele is recessive. So for females, this 75% of the female offspring will not have beards because they are carrying what is for them the dominant unbearded allele. Only this 25% of the females will have the beard. And so 25% of the females will be bearded. Same cross, same genetics, same inheritance pattern, but the alleles show a different dominant and recessive characteristic. So this is the cross as it's shown in the textbook. Everything I just said is what's being shown here, the difference between the males being bearded and the females being bearded. So those are sex-influenced traits. Sex-limited characteristics are extremely similar, just a little bit more extreme form of sex-influenced. Here, regardless of the genotypes, one gender and only one gender of the species is capable of expressing the trait ever. We're going to use as our example here the feathering on roosters. Rooster feathering is our example. Now, I want you to keep in mind as we go through this that these traits are not due to genes that are contained on the sex chromosomes. These are genes on autosomal chromosomes. But for sex-limited traits, we have zero penetrance in the non-expressing gender. In other words, if the female can't express the trait, she will not express the trait. She has the genotype for the trait, I don't care. She can't express the trait. She's homozygous for the trait, I don't care. There's zero penetrance. She will never express the trait. Some roosters, male chickens, not all of them, but some male chickens express what's called the cock feathering trait. And this is your typical kind of classy-looking rooster who likes to strut around the yard. This is the foghorn, leghorn feathering pattern. Only males show this feathering pattern. If you see a chicken that looks like this, it's a male, guaranteed. No hens show this trait ever. Zero penetrance. Regardless of their genotype, they will never show this feathering pattern. The other phenotype for this trait, the other flavor that the feathering comes in, is called hen feathering. Looks like this, your kind of standard boring chicken. And hen feathering is not limited to hens. Hen feathering can be expressed by hens, female chickens, but male chickens as well. The cock feathering phenotype is recessive in male chickens, so it's still recessive, but it is only expressed in males. It is sex limited. So females can be homozygotic recessive, but they don't show the trait. They still show the hen feathering trait. Why is this behaving this way? What makes the zero penetrance in the female? We still don't know. We still don't know the physiological basis for this. We just understand the genetics, such that they are. There are some sex-limited traits in humans, believe it or not. The one discussed in the text is called precocious puberty. It occurs in males. Precocious puberty is a dominant condition. It's very, very rare. The dominant allele is rare in the human population, so it's very, very infrequent that you find someone with precocious puberty. But for a male to have this condition, they only need one allele for it because it is dominant. What do we mean by precocious puberty? Well, the word precocious means ahead of its time, essentially. So precocious puberty is the early onset of puberty in males. Again, only males show this trait. And we mean early. Males with this condition develop secondary sexual characteristics, a deepening of the voice, body hair, etc., at around the age of 4. Now, that seems to be the only manifestation of this condition. Other than that early onset of puberty, these individuals grow up to be completely normal. They're usually a little bit shorter than average height due to the premature stoppage of the growth of their long bones. But other than that, there's no other manifestation of this. But the allele is rare. Since the allele is rare, it's very, very uncommon to find a homozygote dominant individual. Most males that have this are heterozygotes. So now imagine the cross. An afflicted male, we mean a male that had this condition, that showed precocious puberty. An afflicted heterozygous male mates with the much more common homozygotic recessive female. And the offspring from that cross, this is a typical monohybrid cross, heterozygote by a homozygote, recessive, this is a test cross essentially, will have half the sons affected. Right? So we can do that Punnett square, we can look at the data that's in this figure. When we do this mating, half of the kids are going to be heterozygotic and half of the kids are going to be homozygotic recessive. But this is a sex-limited trait. So half the sons will show the condition because they got the dominant allele from their father. Half the sons will not show the condition because they're homozygotic recessive. None of the daughters are going to show precocious puberty. You have homozygotic recessive daughters, but even the heterozygotic daughters with the dominant allele will not show precocious puberty because this is sex-limited, and the penetrance in females is 0. You can do the reciprocal cross, right, where we flip the genotypes of the mother and the father. So now you have a non-afflicted heterozygotic female, non-afflicted because 0 penetrance in females. And she mates with a male that doesn't have the condition because the male is homozygotic recessive. So here neither parent shows the condition at all. But in this mating, half the sons will have the affliction because half the sons got the dominant allele from the mother. Half the sons will not have it because they got the mother's recessive allele. None of the daughters will show the trait. Did half the daughters get the dominant allele from the mother? Yes, they did. But 0 penetrance in females. Period. Sex-limited. Moving on with weirdo things. We'll talk a little bit now about cytoplasmic inheritance. We're actually going to leave the nucleus for once in this class. Mendel's principles, and in fact everything we've been talking about up until now, has applied to nuclear chromosomes. Chromosomes of DNA found in the nucleus. Mendel didn't know that at the time, of course. We didn't even know what chromosomes were. We were just starting to scratch the surface on them. But still, that's what his theories and laws discuss. And even the weirdo cases we've been talking about up until now, that includes things like incomplete penetrance, incomplete dominance, recessive epistasis, as well as sex-limited traits and sex-influence traits. All of those, whether they've been sex-linked or autosomally linked, have been involving nuclear genes on nuclear chromosomes. But actually, not all of your DNA is in your nucleus. I'm pretty confident that all of you know what mitochondria are, the powerhouses of the cell, the ATP-making factories in your cell. How many of you know where mitochondria came from? This is the symbiotic theory. Mitochondria actually came from the ingestion of a bacteria by a very large and pre-eukaryotic cell. So what happened way back when is a eukaryotic cell was hungry and it engulfed a bacterial cell to eat. But for whatever reason, that bacterial cell that was engulfed was not digested. Instead, the bacterial cell kept doing what it knew how to do, which was make ATP. And the ATP highly benefited the engulfing larger cell. So the larger cell was making out on this deal because the larger cell was getting ATP from its not digested bacteria. But the bacteria was winning out too. The bacteria just found itself a very safe place to live where nothing else would ever eat it. And so this was a symbiotic relationship where the bacteria made ATP for the larger cell and the larger cell gave the smaller cell protection. Now there are implications to this. That smaller cell, when it was engulfed, had its own chromosome, had its own DNA, had its own genome. And that genome has stayed with that engulfed cell up until your own cells now that are in your body. So mitochondria have their own genomes. Mitochondria have their own DNA that exists within the mitochondria. This is the leftover remains of that bacterial genome that existed in that free-living cell that was engulfed millions of years ago. So mitochondria have their own DNA. The mitochondrial genome is very small. It's circular, like all bacterial genomes are. And most importantly for this lecture, it is cytoplasmically inherited. The same can be said of chloroplasts in plants. Chloroplasts in plants arose the same way, through engulfment of one cell by another. Chloroplasts have their own genome. Chloroplasts are cytoplasmically inherited, but we won't talk too much about chloroplasts. Now dig into your memory banks for pre-exam material and think about oogenesis and spermatogenesis, the creation of gametes in males and females. Remember I showed this picture? And we made the point that the female egg is so much larger than the male sperm. We made the point that the male sperm is really nothing more than a delivery capsule of male DNA. that the female cell is the nourishing cell, that the female cell is actually a real cell that becomes fertilized, where the male gamete, the male sex cell, really isn't a cell at all. It's just a package of DNA. Given all that, where did your mitochondria come from? Think about it. Think of you. Think of the single-celled you, just conceived. Your father donated half a genome to you. That is the male contribution to new life. Your mother donated half a genome to you. But that first cell, your first cell, the first cell that was you, was actually your mother's cell. Your mother gave you that cell. It was her cell, and she donated it to this new life. So your mother gave you your cytoplasm. My mother gave me my cytoplasm. Your mother gave you your mitochondria. My mother gave me my mitochondria. So this falls under the umbrella of maternal inheritance, things we inherit only from our mothers. My genome is a 50-50 blend of my mother and my father's DNA. 50-50, right down the line. But my cytoplasm, my mitochondria, my mitochondrial genomes, the DNA in my mitochondria, that's all my mother's. If you think about this in terms of a family tree, you can make some pretty profound observations. My mother's, no need to get upset about this. This isn't a tearjerker. This is just the way it is. But my mom's mitochondrial lineage dies with me. I'm a son, obviously. I'm an only child. I have no brothers or sisters. My mom is not an only child, but my mother is the only girl of 4 children. So my mom's mitochondrial DNA came from her mother. But my mom has not passed that mitochondrial DNA on to anyone but me. And I am not female. My kids have my wife's mitochondrial DNA. So that lineage of maternal inheritance has ended on this line. But let's consider my kids. Well, my mother-in-law's mitochondrial lineage lives on. Right? She passed it to my wife. My wife's mitochondria are my mother-in-law's mitochondria. They are the same. They came from the same place. My daughters sparked into life in my wife's cells, my wife's egg that contained my wife's mitochondria. And then, since they're daughters, my daughters will pass that mitochondrial DNA on to their kids. So that's a maternal lineage. This is a pedigree. This is kind of a genetic family tree. We're going to talk about pedigrees at length in the next lecture. But I'm going to give you a few quick pointers just so this makes sense as far as maternal inheritance goes. In all pedigrees, squares are males and circles are females. A line between a male and a female indicates a mating event, and a line coming down shows their kids. The color codes here refer to the mitochondrial DNA, unique mitochondrial DNA. So I'll talk you through this. Here is a mating between a man and a woman. The woman has blue mitochondrial DNA for the sake of argument here. She passes it to all her kids. All her kids are her kids. They were all conceived in her egg cells, so they all have her mitochondria. This son has her mitochondrial DNA, as do these 2 daughters. The son marries a woman, has kids. Well, we don't know if they're married, I guess, but they have kids together. But since he's a male, he doesn't pass that mitochondrial DNA on at all. That lineage has ended on this line. The kids get the mother's mitochondrial DNA because all kids get their mother's mitochondrial DNA, period, end of story. This woman here received her mother's blue mitochondrial DNA. She marries a man. They have 1 child. It's a son. The son has the mother's mitochondrial DNA. He marries, has children. Since he's a son, the kids get his wife's mitochondrial DNA. This central line actually dictates my family history best, I think. This is my mother-in-law. My mother-in-law has her mitochondrial DNA. She passed it to her daughter. Now, my wife is not an only child, but for the sake of argument in this case, that's what's being shown. The mitochondrial DNA gets passed to her daughter. My wife and I get married. We have 2 daughters of our own, and they receive my mother-in-law's mitochondrial DNA, which they will then pass on to their children if they choose to have them. So this is a maternal lineage that lives on. Keep in mind that my mother-in-law's mitochondrial DNA came from her mother. And so those mitochondria are passed on from generation to generation only through the women. So both males and females inherit cytoplasmic traits. I inherited cytoplasmic traits from my mother. I have mitochondria in my cell, and they came from my mother. But since I'm male, I can't pass that DNA on. Only females pass cytoplasmic traits on to the next generation because only females donate cytoplasm to the next generation. Now let's bring this full circle back into genetics. Imagine that mitochondrial DNA is mutated. That's what mtDNA stands for, mitochondrial DNA. Imagine that mitochondrial DNA is mutated, and that mutation leads to a disease. Guess where that disease came from? It came exclusively from the mother, because the mother gave that mitochondria to the child. We already said it, you already knew it, but mitochondria are the powerhouses of the cell. They make ATP, so most mutations in mitochondrial DNA affect metabolism, affect the generation of ATP. There's a fairly rare disorder, but it's metabolically based, called Leber hereditary optic neuropathy. Oosh, not my cup of tea here. LHON. This condition, LHON, causes rapid vision loss to the point of blindness in both eyes. Patients are usually blind between 20 and 24 years of age. This condition is cytoplasmically inherited. It is caused by the death of cells in the optic nerve due to a metabolic disorder in the cytoplasm. So this disorder is passed from an inflicted mother to all of her children, and that's the only way it's passed. The mother is afflicted. Every single one of her children will be afflicted. Amazing, no? In this case, the father is afflicted too. The brother is afflicted as well. They both got, this woman and her brother, both got this disorder from their mother. But when this brother has children, a son and a daughter, he passes it on to none of them because his wife is healthy and doesn't have the disorder. It is only passed from mother to child. However, this woman who develops LHON passes it to all her children regardless of their gender because it is cytoplasmically inherited and passed to all children. The daughters will pass it on to all of their children. The sons will pass it to none of their children unless their wife has this disorder too. No dominance, no recessivity, no multiple alleles. If mom has it, kids get it. If mom has it, sons have it. If mom has it, daughters have it. Everyone gets it from mom. Hope it makes sense. It's a good point at this point in the lecture, maybe to pause the lecture. We've gone over a few different things that are related, but not the same. It's easy to get these different things confused because they share similarities, but they also share differences. So take a moment, make sure this is clear. If it's not clear, make a note of it, something you want to include in your reflection. If we were doing this in class, I would be asking you if this is clear, looking for head nods. So I would like you to be sure that you're comfortable with this material at this point before you move forward. There's another phenomenon called genetic maternal effect. This phenomenon looks just like cytoplasmic inheritance. Things are inherited from mom and mom only, but it has nothing to do with mitochondria or cytoplasm, so don't get these confused. In genetic maternal effect, you have nuclear encoded traits that are found on the normal genome of the nucleus. But the phenotype of the offspring is directly determined by the genotype of the mother. It's worth saying that again. For genetic maternal effect, the maternal genotype dictates the offspring's phenotype. It's almost like there's a single generation lag in the manifestation of the phenotype. Most traits that demonstrate genetic maternal effect require some maternally provided substance or molecule or protein that's present in the egg during the early development of the organism. And so it's the mother's genome that makes this substance. So it's the mother's genome that influences that trait in the offspring. It gets a little abstract. Let's go to an example to see if it kind of drives the point home. We're going to use snail shell coiling as our example. Most snail shells coil to the right. This is a clockwise coiling. It's also referred to as dextral. But sometimes you find a snail shell that coils to the left, counterclockwise, or sinistral, because left is sinister and lefties must be evil. And I hate that stuff. Anyway, dextral coiling to the right is dominant. That's why we see more of it. So we can use the S plus denotion to show the dominant allele, and we'll use just plain S to denote the sinistral, left-handed coiling. So far, pretty simple. Sounds like a single gene trait. Mendelian principles, blah, 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 blah. But we're talking about genetic maternal effect. So the direction a snail's shell coils in is determined by that snail's mother's genotype. What? All right. So, here's a dextral male who's homozygotic dominant, S+, coils to the right, mated with a sinistral female, SS, homozygotic recessive, coiling to the left. You look at the offspring. The offspring is genotypically heterozygotic with a dominant allele. Phenotypically, left-handed coiling. Recessive phenotype, dominant genotype. Why? Because the offspring phenotype reflects the maternal genotype. Because the mother was homozygotic recessive, the child shows the recessive trait, irregardless of their genotype. Now you do a self-cross, right? The brothers and sisters of this F1 are mated. Think about that for a second. That's going to be S plus S, heterozygotic, mated by S plus S, heterozygotic. Hopefully you can do these in your sleep by now. Heterozygotic by heterozygotic cross is going to give us 3/4 dominant, 1/4 recessive. Typical Punnett square. Look at the offspring of that cross, and they are all dextral. Well, keep in mind that the phenotype in the offspring always reflects the maternal genotype only. So the offspring are 100% dextral because the mother had the dominant genotype. She didn't show that genotype because she was lagged due to her mother, but she had a dominant allele, and so all her children show the dominant trait. Some of those children are homozygotic dominant genotypically. Some are heterozygote. Some are homozygotic recessive. Yes, but they all show the trait that the mother's genome encoded. These females, when crossed with anyone, are going to have 100% sinistral offspring because that's what this genotype encodes. These mothers, these females, when they are crossed out, regardless of who they're crossed with, will have all dextral coiling offspring because that's the genotype they have. So it's this lag from one generation to the next. So again, if we go up to the top to say it in words, we have a homozygotic dextral male crossed with a homozygotic sinistral female. All of the offspring are genotypically heterozygote, genotypically dextral, but all of the offspring have sinistral coiling shells because that's what their mother's genotype encoded. You do the self-cross, you should get 3-to-1 dominant to recessive, but what we see is all dextral because that's what the mother's genotype encoded, the dominant trait. I really like the idea of saying this skips a generation because it really kind of sort of does. What we see in the offspring is what the maternal genotype reflects. Another phenomenon, genomic imprinting. Mendel tells us that reciprocal crosses don't change a thing. We've talked about reciprocal crosses a few times, but just to put the fine point on the definition, this is when you change the genotype and presumably phenotype of the two parents. So if you started with a mating that was a short-legged female with a normal-legged male, the reciprocal cross is the normal-legged female with the short-legged male. Okay, no real difference. The genders switch and the phenotypes don't, or vice versa, the phenotypes switch and the genders don't. This works most of the time because according to Mendel's rules, it doesn't matter which chromosome or which allele is given by the mother and which is given by the father. What matters is that you have two alleles and one is dominant over the other. Well, this isn't always true. The expression of some genes, the expression of some individual alleles, is sometimes, rarely, but sometimes strongly affected by which parent that allele came from. Interesting. This differential expression of alleles that depends on the parental origin of that allele is called genomic imprinting. I'll give you an example. There's a human gene called IGF-1. It stands for insulin-like growth factor. It plays a role in metabolism, but it does other things too during development. We get 2 alleles for IGF-2 for this gene, one from mom and one from dad. That's true of any gene that we find that's on an autosomal chromosome. But there's genomic imprinting at work here. Mom's allele for IGF-2 is always silenced during development, regardless of whether it's the dominant allele or recessive. If it's maternal, it's silenced. While the paternal allele that came from dad, regardless if it's mutated or healthy, dominant or recessive, is the one that's expressed in the fetus and placenta. Without a paternal allele, even if the maternal allele is there and wild type and healthy, without a functioning paternal IGF-2 allele, we see the effects of no IGF-2, low birth weight, small placenta, because the father's allele is the only allele that can be expressed during development. Interesting. Why would that be? You might be wondering how that is too, and I can explain how in later lectures. We'll talk a little bit about how imprinting works. I'm going to save it for later lectures though, because I don't want to overcomplicate it. If you're dying to know, you're more than welcome to come to office hours, and I can go into some of the molecular biology of that for you if you'd like. But the why is really interesting. Why does this happen? Why would you always silence the maternal IGF-2? Why the maternal IGF-2 and not the paternal? Well, IGF-2 plays a role in nutrients, it plays a role in metabolism, insulin-like growth factor, etc. It plays a role in placental development. And you see the paternal IGF-2, coming from the father, is a more high-powered allele, almost always. And it directs a larger placenta that quite literally steals more nutrients from the mother for the fetus. Really what this paternal allele is saying is, I want this child, I want my child fed at all costs. In a genetic, physiological way, the paternal allele is willing to have a negative effect on maternal health, right? The theft of nutrients. In some ways, a developing fetus is a parasite. It's stealing nutrients from the host. The paternal IGF-2 allele is allowing the developing fetus to develop at the expense of the mother's health. I want my child fed at all costs. The maternal IGF-2 allele is not quite so vehement. The maternal IGF-2 allele does promote placental growth, but not at the same level. And so it limits the maternal nutrients to the fetus. The maternal IGF-2 allele can be thought of as saying, yeah, I want my child fed too. I'll build a placenta, but not at the cost of my own health. I, as a maternal organism, want to be able to live healthy enough to be able to breed again and get my genes into the next generation another time. These are competing objectives of these two alleles. The paternal allele says, to hell with your current health, you feed this child at any cost, big placenta, lots of nutrient theft. The maternal allele says, I want the child too, I want to feed the child too, but it's okay if the child doesn't develop quite as fast and quite as well and quite as healthy, because I don't want to give as many nutrients of my own to that developing fetus. Which of these two alleles do you think the fetus prefers? Now let's get away from the touchy-feely, lovey thing of kids and mothers. We're talking about a living entity versus another living entity. You better believe that the fetus wants the paternal allele on. From the fetus's perspective, that paternal allele sounds really, really good. And there was an evolutionary survival pressure to keep dad's allele on, get that big placenta, develop big and healthy, and turn off mom's slightly attenuated allele that wasn't quite ready to give up so many nutrients. And so that's what happened. Evolutionarily, mom's allele was always turned off and dad's allele was kept on. Now, that's a mechanism. So if dad's allele is somehow mutated and won't promote placental growth at all, the fetus has no way to sense that. This isn't a conscious decision. These are biological mechanisms. So mom's allele is always turned off and dad's allele is always kept on. Is that always 100% of the time in the best interest of the fetus? No. But playing the odds, dad's allele is much more likely to be better for the fetus than mom's. And so that's the gamble that the genomic imprinting plays. There's paternal imprinting and maternal imprinting, so you can imprint from either parent. Sometimes the maternal gene is the only gene that's expressed. The maternal allele is the only allele that's expressed. But the idea of a gene being kept on from a particular parent and not the other parent, that is genomic imprinting. And without fail, of course. The allele that's kept on is usually the allele that best helps the survival of the individual keeping that allele on. It's every man for himself out there. This is referred to as the genetic conflict hypothesis, and for good reason, because it is conflict. It is opposing wishes, opposing strategies at that genetic allele level. Really, really interesting stuff. Some other examples of genomic imprinting. Prader-Willi syndrome and Angleman syndrome or Angelman syndrome are genetically identical. So they are based on the same exact genetic condition, but they are phenotypically different. Both of these are caused by identical deletions of a very small region on the long arm of chromosome 15. So we have our chromosomes numbered, we have 22 of them autosomal, and then the 23rd pair is the sex chromosomes. Well, there's a very, very small deletion on the longer arm, the longer segment of chromosome 15, and that results in both Prader-Willi syndrome or Angleman syndrome. So how do you know which one you have? Well, from a diagnostic point of view, kids with Prader-Willi syndrome present as short. They usually have cognitive defects. They usually have very small hands and feet. They suckle poorly as infants, and so they usually have low infant weight, but then they become obese as kids. Kids with Angleman have a very different presentation. They exhibit frequent, unprovoked, and uncontrollable laughter, uncontrollable movements of their muscles. They usually have a larger than average mouth size, and they're prone to seizures. Very, very different presentations, same exact genetic defect. What gives? Well, Prader-Willi is due to an inherited chromosome 15 deletion from dad. If dad's chromosome had this deletion on chromosome 15, you have Prader-Willi. Angleman is when that deleted chromosome 15 is inherited from mom. So what affects the syndrome is only the parental origin of the deletion. Some of you may know, some of you may not, but Colin Farrell's child has Angleman. His son has Angleman syndrome. And, I'm not into the blame game thing here, and this is not what it should come down to in the home environment. But genetically speaking, that Angleman in this child did not arise from any defect in Colin Farrell's DNA. Because Angleman is due to a chromosome 15 deletion from the mother exclusively. Had this son had Prader-Willi syndrome, then we would know definitively that the chromosome 15 deletion came from Colin Farrell himself. Why this works this way, no one has any idea. And more than anything we've discussed, this flies in the face of Mendel. Mendel says that reciprocal crosses should have no effect whatsoever. Mutations, whatever their effects might be, dominant or recessive, whether they come from the mother or father, should not play a role, but yet it does. We'll talk a little bit about epigenetics and anticipation, then we'll start wrapping up the lecture from there. Epigenetics is this idea of standing on top of genetics or trumping the inheritance patterns, something that affects the inheritance of a trait and the manifestation of a trait even more than the DNA and the genome. Epigenetics is what explains genomic imprinting. DNA is a molecule, and as a molecule it can be altered or modified in ways that are reversible. So yes, something can be done and then undone, but also silencing. We call these epigenetic marks. And when we say silencing, we mean that DNA is wrapped up so tightly that it becomes virtually inaccessible. And because it's inaccessible, you don't express that gene. Going back to the idea of IGF-2, we can epigenetically mark the maternal allele, causing it to be silenced or wrapped so tightly that it will never be expressed in the fetus. The fetus has no choice but to default to the only allele for that gene that is not silenced, and that's the paternal allele. Some of you may know the story about bees. There's only one fertile female bee in a beehive, and that's the queen bee. All of the baby bees come from that one fertile queen bee. So how do you make one fertile bee and all the other female bees are sterile worker bees? How do you do that when every bee in the hive is genetically identical? So they have the same exact genome, the same exact alleles, but one of them is large and reproductive and the others are smaller and non-reproductive? How can that be? Well, something must be standing on top of the genetics. And that's the epigenetics, that's silencing. By silencing certain genes in the sterile beads and doing it with royal jelly, doing it chemically, doing it with an environmental component, you can silence the genes that make you a worker bee, thus leaving only the fertile queen bee genes to be expressed. Anticipation. Anticipation is when a trait becomes more strongly expressed or expressed earlier in life in subsequent generations. Very interesting. So here we have the age of onset idea. Now keep in mind that the genotypes of these children, these people, are the same for this disorder. So the grandparent, the parent, and the grandchild all express the trait. But the grandparent expressed the trait in adulthood. The parent expressed the trait in childhood. The grandchild expresses the trait in infancy. Same trait, same genotype, different ages of onset. It also works with the severity of the trait. Again, same genotype in 3 generations. The grandparent showed the trait, but it was mild. The parent showed the trait, but it was moderate. The grandchild shows the trait, and it's severe. So in subsequent generations, from grandparent to parent to child, the trait either expresses earlier in life or more severely. Genotypically, these individuals are the same. Phenotypically, they present differently. Why? Don't know. No idea. We are just now scratching the surface on understanding epigenetics, how it works, what is the physiological basis of it. We're just now starting to understand this phenomenon and how it interplays into anticipation. But there's much more left to do than been done already, and we most certainly do not understand these concepts thoroughly. Let's wrap up with some other environmental effects on phenotype. Now, the royal jelly story certainly is one of those examples. And we've already talked a few times in this course about nature versus nurture, and we will again. The idea of penetrance, those cold bunnies and their black ear tips, etc. So we have seen how the environment affects phenotype before. These concepts address the roles played by the environment in the manifestation of phenotypes, but let's talk a little bit more about the role environment plays and when that causes an exception to Mendel's rules. Mendel says that 2 individuals with 2 identical genotypes should be identical because it's all about the genes, but we know that's not necessarily true. The textbook says, and I'm paraphrasing a bit, the genotype sets the potential, the potential for the development. But the phenotype, that is the development of the trait, develops within the confines imposed by the environment. So the genotype sets the stage for the trait. It's then the interplay between the environment and the genotype that makes the phenotype develop. Here's an example. There is a vestigial wing mutation in fruit flies. That mutant allele is called VG for vestigial. It's a mutant. It's recessive. When you rear flies or grow flies at temperatures below 29 degrees centigrade, you can show the recessive trait, the vestigial wings, in the homozygotic recessive fly. But if you take those same flies that have that mutant genotype and should express vestigial genes, vestigial wings, but you grow them at temperatures of 31 degrees centigrade or higher, they develop normal wings. A wild-type non-mutant fly will have normal wings no matter what temperature you rear them at. So most certainly, the VG mutant genotype set the stage for vestigial wings. But it was the interplay of the environment, in this case temperature, and the genotype that manifested the phenotype in these flies. Phenylketonuria, some of you may have heard of that in another class, PKU. It's a genetic disorder. It causes mental retardation in individuals with PKU. The cause of this is that people with this disorder cannot metabolize the amino acid phenylalanine, and so very early on in life there's a massive accumulation of non-digestible phenylalanine in these individuals, and it causes brain damage. So it's the toxic buildup of phenylalanine that causes the disease. It is genetically based that an individual have phenylketonuria. So the inability to metabolize phenylalanine is genetically based. But the syndrome, the disorder, the mental retardation comes from the toxic buildup of phenylalanine. So we now have a way to diagnose PKU at birth. And immediately, once diagnosed, soon after birth, if an individual is placed on a low phenylalanine diet, they will never get to the toxic buildup of this amino acid, they'll never get the brain damage, and so they'll avoid the disease. By manipulating the environment, we can manipulate the phenotype. And incidentally, you may have seen these warnings on labels before. Attention phenylketonurics, contains phenylalanine. This is on many processed foods. This is because if somebody with PKU is to consume this food, they run the risk of brain damage, a toxic buildup of phenylalanine. But if they stay away from foods that are high in phenylalanine, they'll never get the disorder. Sometimes, in the most extreme case, the environment alone can mimic a mutant trait. The environment alone can give rise to a nearly identical phenotype to a particular mutant genotype. One example is the condition of eyeless in flies. In fruit flies, there is a recessive allele called eyeless that, when an individual is homozygous for it, causes very small eyes to form in the individual. Here's a normal fly on the left, here's a normal eye, fruit fly eye, and you can see the individual on the right has virtually no eye at all. This is a genetic mutation. This is an individual that has two recessive eyeless alleles, mutant alleles. If you take a wild-type fly who's homozygotic dominant for this, should have normal eyes, but you expose that wild-type fly to sodium metaborate, a chemical, you're going to have the eyeless phenotype manifest. The individual fly that is genotypically wild-type will develop the recessive eyeless phenotype when exposed to this chemical, because the chemical mimics the recessive phenotype. This is called phenocopy, when an environmental stimulus causes the copying of a phenotype in the individual, regardless of what their genotype is. I'm going to end with the idea of continuous characteristics. We're about to move away from the world of Mendel for a while, completely in fact. We're going to go on to pedigrees, and then after that we're going to talk about linked genes. It's going to be a while before we come back to these ideas. So before we close the book on Mendel and transmission genetics for a while, I'd like to get something straight, something I've kind of been misleading you about all this time. There are such a thing as two phenotype traits. Yes, peas can be yellow or green. They can be round or wrinkled. Pea plants are either tall or they're short, etc. But the vast, vast majority of traits out there are not two phenotype traits. They are not A or B, yellow or green. Human height is the perfect example. Humans are not tall or short. Humans are 6'4", 6'3.5", 6'3", 6'2.5", 6'2", 6'1.5", and even finer increments between those boundaries if we choose to measure to that precision. These are called continuous characteristics, and most traits in most animals are continuous characteristics. This is when there is a full spectrum of phenotypes, from the shortest human being ever measured to the tallest human being ever measured, and every single height in between. What makes a trait continuous, we also use the term quantitative traits for this. So we're going to end this semester with a discussion of quantitative trait loci, and that's where this is heading. That's what this is setting the stage for. What makes a trait continuous, what makes it quantitative, what makes it that it has to be measured. Not green or yellow, how green, how yellow, not tall or short, how tall. Let's measure the height. What makes these traits continuous is that a single trait, like height in humans, is controlled by many genes, and oftentimes those genes interacting with the environment. Because there are so many different inputs to the trait and so many different combinations of genes and environment, you get every single possible flavor of height across the spectrum. Traits that are governed by many genes are called polygenic characteristics. Many genes, polygenic. And the same is true in reverse. There are some traits out there where one gene, in its own singular way, can control and influence multiple different traits, and we call this pleiotropy. Pleiotropy is one gene controlling many traits. Polygenic is many genes controlling one trait. We've already talked of an example of pleiotropy, actually, and we didn't even call it that. This yellow fur color is a pleiotropic trait because the gene controls fur color and death. Remember, this one gene, in its heterozygous state, gives a yellow-furred mouse. In its homozygous dominant state, leads to cell death. Those are two different traits, if you ask me. Yellow fur and death, two different traits, but encoded and controlled by one single gene. So what did we talk about today? Well, we talked about quite a bit, and we kind of threw a potpourri together as well. A lot of terms, a lot of similar concepts that were subtly different. We started off talking about sex-linked characteristics in the last lecture. Don't confuse sex-linked with sex-influenced or sex-limited. Sex-linked characteristics are found on the X chromosome. Examples of these are color blindness. Sex-influenced characteristics are characteristics that have different penetrance in the two different genders of a species. These are autosomal genes found on autosomal chromosomes, but they're more readily expressed in one gender versus the other. We use billy goat beards as an example. We then moved on to sex-limited traits. Sex-limited traits have zero penetrance in one species, in one gender versus the other. Still autosomal, still not on the X chromosomes, but the expression is limited to one gender and one gender only. We used the chicken feathers as an example there. We went on to talk about genetic maternal effect and cytoplasmic inheritance. Genetic maternal effect, we used snail shell coiling as an example, where there's this generation lag between genotype and phenotype. The phenotype expressed by the offspring reflects the one genotype of the mother. Cytoplasmic inheritance was our LHON, the Leber optic, whatever, neuropathy thing, LHON. That's where we had a mitochondrial-based trait, so a trait that's encoded on mitochondrial genes. Since you get your mitochondria from your mother, this trait is passed on from mother to child exclusively. And then we talked about genomic imprinting and the genetic conflict hypothesis. We used IGF-2 as our example, where the maternal IGF-2 is shut down completely, and the paternal IGF-2 is left on because it usually promotes fetal development more vigorously. So the genetic conflict hypothesis basically states that the needs of the fetus trump all. And if the needs of the mother and the needs of the father are in conflict, the needs of the developing fetus will win. In this case, the paternal genes are allowed to stay on because they promote fetal growth. We talked a little bit about epigenetics, standing on top of genetics. Here, molecular modifications of DNA change that DNA, silencing some alleles and allowing others to express. It is epigenetics that explains the genetic conflict hypothesis for IGF-2. We defined anticipation when one trait is expressed more strongly or expressed earlier in life than subsequent generations. We talked a little bit about phenocopy and the other role played by environment and manifesting of phenotypes, influencing penetrance, etc. And we introduced the idea of continuous or quantitative characteristics. Here there is not A or B, tall or short, but the full absolute spectrum of all phenotypes in between the two extremes, from very short to very tall with every height in between. We defined the terms polygenic, meaning many genes affect a single trait, and we also defined the term pleiotropic, meaning one gene affects multiple traits. We're going to move on to pedigrees now, kind of get a little bit into more population-type stuff, a little bit away from Mendel, and talk about some of the applications of these human analyses using pedigrees and some genetic testing that we do on humans and what the implications of all that are. So we'll get to that in the next lecture, but as always, thanks for watching.